Okay, hello everyone at home. No, Google Meet. Okay, you see this, it's too much. And my calendar's still not sorted out, so I have to go look for an email that I sent one of you.
Mama, are you teaching the grade 8 online?
No.
You teach grade 8?
I'm doing grade 9 and 10 and you guys. Okay, so Lizandia, I saw one from you. You accepted. It's nice when you accept it because, then I can look in, because then if one of you accepts, then I can easily find, because I can't get the link on my phone. I don't even have time to figure it out. Okay, so let me accept Google Meet. Yeah, so if you do that every time, then I know I can quickly find it and put it on.
Yeah, because otherwise the notification keeps coming up to say everyone has to try and accept.
Yes. So I wonder if anyone's here. I'm the first one here. Maybe just tell them we are here.
I'll send a message on.
Okay. This thing I need to fix. It's not working so well anymore. Yeah.
But most
Sort
of
of
us
okay.
did the same stuff.
Last science. Yes. What is your name?
It's funny, the same people who do
Okay.
science too.
All right, so let's look at page 126. And do you have break after this?
No.
I was going to say, you can go thaw in the sun afterwards. It's freezing in here. Okay, so, yeah, I don't want to really do all this on the board. All right, so figure 1. So this is activity 5.13, page 126. You've got 3-point mutations, A, B, and C. So which kind of mutation is present in A?
Insertion.
All right. So that G was inserted, and then could you see how the codons change after G? So the codons for mutation A would be TTA, CCG, then CTA, GCA, GGA, and then there's an extra one. So what is that called now when the codon sequence, or not the sequence, the actual codon placement changes? A frame shift. And it's an insertion, not addition. The textbook says addition, no, it's not maths. It's an insertion mutation. Did anyone use the DNA coding strand to see what that coding strand normally codes for in terms of amino acid sequence? Okay, but you understand the concept. All right. So did you use page 17 to work out which amino acids are coded for by mutation A DNA? Did you? Do you understand it though?
Yes.
You're supposed to just tell me which amino acids are coded for. You see? Oh, but then you first got to work it out to messenger RNA. So, okay, so then you can do the rest yourself. So if that's TTA, CC, it was CCC, but, oh wait, no people. Okay. Oh, it is an insertion. Sorry, I didn't see. The G is put in there. Yes, it is an insertion, then TAG, etc. All right. So there were the codons, original, but now the codons are going to change. So you might be asked to show that those CCG becomes the new codon and CTA, not TAG anymore. So they might ask you to copy it into mRNA. So if we take the mutated one, well, hang on. If we take the original DNA, all right, what is it copied into in terms of mRNA? AAU, and then CCC? CCC becomes G. No, let's just do the black, the original. The red is going to be the mutated A. And then TAG? Okay, please just go to page 17 so that practice for the test that's coming up. Go have a look at page 17. I won't ask you to do the whole string. They might give you a short question like that.
And which test is this?
The cycle test.
Oh.
and your prelim and your November. I'm thinking anything. Okay? So what does mRNA, what does the codon AAU relate to? Sorry? Aspartame or something, okay? ASN, asparagine, it doesn't matter. GGG? Is it? GGG is? Did you all see glycine? GLY. Okay, as long as you can find them. And then AUC? Isoleucine, okay, you don't need to know the name. But now if there is mutation A, in other words, guanine base is added, guanine nucleotide is added to the DNA, now all these codons shift. So now TTA is still copied into AAU, but what is CCG copied into? mRNA. G, okay, and then now it's CTA, the next codon. So it's going to be copied into messenger RNA. Okay, go and find, okay, so AAU will still code for asparagine amino acid, ASN, doesn't matter what it's called. So now go look up GGC. What amino acid will be brought in by the tRNA? Sorry? Okay, glycine again. Alright, but the next one? Asparagine, okay? ASP. Alright, so it's different. So maybe because now all the next amino acids are going to be different from the original, from here. So what do you think will happen to that protein when the polypeptide chain folds up? Sorry? It's different, and therefore maybe? It's mutated. The DNA is mutated, the protein is now different, so maybe it doesn't work. Okay, so maybe it doesn't fold up properly, and maybe all the enzyme active site is wrong. It won't work. Okay, so insertion, because it changes all the the codons on the DNA after the mutation, it results in a drastic change to the resulting protein, and therefore maybe it won't work. Okay, is anyone on here? No? It's just me? Okay, it's fine. They can watch the video, it's fine. Alright, so then if we go back, so now mutation B is substitution. Okay, let me just rub this off. The reason why I want to do this with you is because you need to be able to answer some kind of question like this. So mutation B, it's TTA-CGC. So that is cytosine, nucleotide is substituted with guanine. Okay, so what will the first codon be copied into mRNA? Stays the same. So it's still AAU. What will CGC be copied into messenger RNA? GCG. All right, go and find out what that's going to be. What amino acid will that be? That one will still be the same. This one will still be the same. Iso, what's the middle one? Alanine. Do you think it's going to make a difference? Maybe, yes. It's the wrong amino acid, so maybe the protein doesn't fold properly, maybe it doesn't work. Okay, but the previous one, I think the second one was still glycine. Even when it was, the mRNA was copied from the mutated DNA. If it was just that mutation where it was still glycine, the protein might still work. But when you go to varsity and you learn about it, then they might say, no, protein won't fold properly. It's called the wobble hypothesis, but we're not going to worry about that. So if the correct amino acid is brought in, but by a different tRNA, because the anticodon is slightly different, it might not have an effect on the protein. At grade 12 level, we say that, because it's still the same amino acid. But now, wrong amino acid is brought in, so therefore maybe it doesn't work. Okay, are we happy with that? All right, so let's look at mutation C is a deletion. Okay. So it was TTA, CCC, and the T was taken out. No, yes, this one. That was taken out. So AGC, and there's a C there. Okay, so now it's a deletion mutation. All right, so once again, the mRNA, first one will stay the same. It's TTA on DNA, will still be AAU on mRNA. ASN, asparagine amino acid, is still brought in. This one, still the same. So you might be asked to write down the mRNA, and glycine will still be brought in. What will it be copied into? mRNA. Next one. Waiting to go. What will this be copied into mRNA? Okay, what amino acid will be brought in? Sorry? Serine. Oh, okay, so I didn't read it just now. So can you see now that every amino acid thereafter is different? So deletions and insertions are going to cause a major change to the protein. Not going to work. A substitution, if the same amino acid is still present, it might work. It will probably work at grade 12 level. All right, but if it's a different amino acid, like mutation B, then so if it was supposed to be glycine, but now so in the new protein, if that one's right, and all the others are right, but this one's wrong, it might not work. Well, I don't think it will work. Okay, all right. Does it make sense? I don't know if I've explained it well enough. All right. So that's question number 2 on page 126. Use your knowledge of the genetic code and protein synthesis to work out the amino acid sequence for each piece of mutated DNA. Which type of point mutation, number 3, will have the smallest effect on the structure of the completed protein? Substitution. Why? Explain your answer. Because
It doesn't result in a frame shift.
there's no frame shift, but they might not know that you know what that means. So maybe just say that there's only one amino acid that is different.
How will they not know that we don't know what it means?
Because, okay, frame shift, that word frame shift, I don't think it's in the SADs. So if you've just heard the word frame shift, but you don't explain what you mean by
If I don't
it.
know what it means, then how did I come up
Okay,
with it?
so I've used the word frame shift so that you know what it means, that all the codons or amino acids thereafter change. But for a better answer, okay, why would it not have a great effect? It's because it's just one amino, which type of mutation would have the smallest effect? Substitution. How many amino acids change? If it's substitution, it's one amino acid that changes. If it happens to be the same amino acid, is the protein changed? No, because the same amino acid is still coded for. But if that new nucleotide that's substituted results in a different amino acid being brought in, then what? Then maybe the protein doesn't work. Just validate. Don't just say frame shift. If you say frame shift, then say, okay, so for the other two, where there's an insertion or a deletion, where there is that frame shift. So you say no. Okay, the protein wouldn't work because after the mutation there is a frame shift, which means that all the next codons will be different. So therefore all the following amino acids will be different, and therefore the protein will not be the same. Okay, rather than just saying frame shift. Okay, I'm just saying that because then I see how they mark. All right, so rather say that. Don't just say frame shift, that after the mutation all the codons change, therefore resulting in all the next amino acids till the end of the polypeptide chain being different. Okay, all right guys. Anything else there? All right, then it said list 4 types of chromosomal mutation, but we don't need to know all of them anyway, so I should have said don't do that. But I've crossed it out. So the one that you could maybe talk about, chromosomal would be like aneuploidy or polyploidy. You mentioned it yesterday, remember? too few or too many chromosomes, aneuploidy, or polyploidy, too many, or many sets of chromosomes. We don't have to do the other ones in the book, which is on page 125 at the bottom. We don't have to do translocation, duplication, bits moving around, whole genes, whole sections of chromosomes moving around. No. We just have to understand aneuploidy, where there could be extra chromosomes or too few chromosomes, like Down syndrome and Turner syndrome and all of those, which you must make sure we just remember. And polyploidy, like in plants. Okay. All right. So, question, activity 514. I must just read it again. So, yeah, so as I'm saying, that's why I wanted to write it here. What is the DNA? They're going to say, maybe. What's the mRNA? What's the amino acid sequence? Okay, now what if this base changes? Now what's the new mRNA? So, how does the amino acid sequence change using that mRNA table? What will happen to the protein? Depends on which type of mutation it is. And then you've got to explain nicely. Okay. All right. Okay, 514. Bacteria reproduce asexually by, through experiment, binary fission. 2 sterile petri dishes are filled with a nutrient agar medium. Red bacteria from a single colony of bacteria growing on another petri dish are smeared onto the agar on both petri dishes. I'm just trying to remember the question. One petri dish A, excuse me, A is placed in UV light, and the other petri dish B is left in the dark. All right, and the 2 petri dishes are examined after 5 days. In A, all but one of the bacterial colonies growing on the agar are red. All but one. So they're saying only one. What? They're all white except one. So one's like that.
Yeah.
Okay, so these colonies have grown. Alright, you remember that from grade. And you will see, hey, there'll be questions about antibiotic resistance and things like that, you know? Especially with genetic engineering, they bring grade 11 stuff in. But we still link that up now. Okay, what hypothesis do you think did the person doing this experiment test? Hypothesis. If this, not if, this, then that. Or if, yeah. If this, then that. Come on, there's UV light there. What is that called? It's a mutagen, alright? So, okay, alright, so UV causes what to mutate? Yeah. UV radiation causes bacteria to mutate. This, UV radiation, then that, causes bacteria to mutate, or not. You could have said UV radiation does not cause bacteria to mutate. It's still a hypothesis. This, then that. Okay. Now, which variables were investigated? So they're talking, okay, what's independent variable? UV light or not? UV light or normal? Bacteria like to grow in the dark, okay? No, they can grow anywhere. Okay, it's just in the dark. So UV light or not, the bacteria exposed to UV light or not, dependent variable of
Bacteria.
the colony, yeah. Okay, changing color of the bacterial colony. All right. Then controlled variables, lots. Which was?
5 days.
So the bacterial colonies were allowed to grow for 5 days. Okay. What else?
Petri dishes were sterile before the bacteria were introduced.
So that there aren't other bacteria there. Anything else? They all had the same nutrient agar, same growth medium, therefore they're going to be, growing the same way. I mean, you could say they were both inoculated in the same way, but we're not going to go there. All right, then, That's 3, sorry. So 2 was independent and dependent, and question 3 is controlled. We separated. What else can I have? Oh, wait, hang on, there's another controlled. That they were both inoculated with was the same. It was the red bacteria, with these little red colonies growing. Do you remember in grade 11, yellow, orange, white colonies growing in your agar plates when you touch them and sneeze them? Okay, so which apparatus? Now this one I've got to think about, and we'll think about it tomorrow. Okay, we'll think about it tomorrow. We've got how many lessons tomorrow? We've got a triple. No, yes, we've got a triple. Well, 1.5 hours. Double. Actually double, but it's a triple now. Yes. Okay, it's like a normal double. Alright, so we'll carry on with that tomorrow. Okay.